Ever feel like you're just drowning in information these days, especially with science moving so incredibly
Oh,
fast?
absolutely. It's tough to keep up, even when you're really interested.
That's exactly why we started the deep dive. Think of us as your shortcut, you know?
hmm
We try to cut through the noise, get you the important stuff.
The insights that really matter.
Exactly, to help you become genuinely well-informed without feeling overwhelmed. So today, we're taking a deep dive into a really foundational chapter. It's from Essentials of Genetics, 10th edition, focusing on genomics, bioinformatics, and proteomics.
A huge area.
It really is. Our mission today, to sort of unpack the whole journey from figuring out the first genetic sequences all the way to, well, the ethics of maybe designing new life forms.
And it's such a timely topic. What's fascinating, I think, is just how fast these fields are moving. They're giving us insights into life itself that were, frankly, unimaginable just a short while ago. We're talking about the omics revolution.
Sure.
Genomics, bioinformatics, proteomics. They're not just changing biology. They're really reshaping medicine, agriculture, even how we think about ourselves.
So buckle up.
Pretty much. Get ready to explore the huge landscapes of DNA, RNA, proteins.
Yeah.
Figuring out how their sequences make them work and how we've learned to read them, and now even maybe rewrite them.
Okay, let's start at the beginning then. The genomics revolution, where did it kick off?
Well, you really have to go back to 1977. Fred Sanger, a brilliant scientist.
What did he do?
He sequenced the entire genome of a tiny virus, FX174. It's only about 5,400 nucleotides. Sounds small now, maybe.
Yeah, compared to a human genome. Tiny.
But sequencing the first complete genome, that was absolutely monumental. It really launched the whole field of genomics.
Okay, so that's a tiny virus. How do you scale that up? How do you sequence something massive, like the human genome?
That's where whole genome sequencing, or WGS, comes in. It's sometimes called shotgun sequencing.
Shotgun sequencing. Why that name?
Well, the analogy helps. Imagine you have a massive textbook, right? Your
Yeah.
genome. You need to copy every single word.
Okay.
Instead of going page by page, which would take forever, you make, say, thousands of copies of the book.
Right, yeah.
And you just blast them apart. Rip each copy into thousands of tiny, overlapping strips of paper.
Okay, chaos.
Seems like it. But then, your job is to piece it all back together. You look for matching sentences, matching phrases on those overlapping strips.
I see. The overlaps are key.
Exactly. That's the WGS process. You take multiple copies of a chromosome, you break them into countless short overlapping fragments, you do it mechanically, or use these molecular scissors called restriction enzymes.
Like E. coli, maybe?
Yeah, like E. coli.
And
A 6-base cutter like that could chop the human genome into, maybe 700,000 fragments.
Wow.
And then powerful computer programs take over. They align all these fragments, they're called contigs, for contiguous fragments based on their identical sequences.
Yeah.
Slowly, piece by piece, they rebuild the entire genetic text.
That sounds computationally intense.
Oh, massively. And initially, people were skeptical it would work for really big, complex genomes, like ours.
But it did work.
It did. The real proof of concept came in 1995. J. Craig Venter and his team at Tiger, they sequenced the genome of Haemophilus influenza.
It's a bacterium, right?
Right. First free-living organism sequenced. About 1.8 million base pairs. That showed shotgun sequencing could handle bigger jobs.
And the tech improved, too.
Dramatically.
Yep.
Computer-automated sequencers, high-throughput sequencing, productivity just exploded.
Okay.
Went up over 500-fold.
Yeah?
And the cost, it plummeted.
How much?
Went from about $1 per base pair down to less than a tenth of a cent, maybe even less now.
Incredible.
And that massive drop in cost, that increase in speed, that was absolutely essential for the next huge step.
The Human Genome Project.
Exactly.
So you sequence this mountain of data, billions of base pairs,
Right.
but what do you do with it? It's just letters, right? A, T, C, G.
That's the critical question. Raw sequence data isn't knowledge. It's like having that ripped up textbook, but no idea what the words mean or how the sentences fit together.
So you need a way to interpret it.
Precisely. And that brings us to bioinformatics. It's really the digital backbone of all of modern genetics.
Okay, define that for us. Bioinformatics.
Simply put, it's using computers, hardware, software, math to, well, organize, share, and most importantly, analyze all this biological data. Gene sequences, gene structure, how genes are expressed, protein structures, functions, all of it.
And it became crucial just because of the sheer volume.
Absolutely indispensable. The amount of data was just exploding. Think about GenBank. It's the biggest public DNA database.
How big are we talking?
Over 220 billion bases. from more than 100,000 different species. And get this, it doubles in size roughly every 18 months.
That's mind-boggling. How can anyone keep up?
Well, that's what bioinformatics tools are for. It's not just storage, it's analysis. Comparing DNA sequences like aligning those contigs we talked about, or seeing how similar a human gene is to a mouse gene.
What else?
Identifying genes themselves. Finding the control switches like promoters and enhancers. Predicting what protein a gene will make and maybe what that protein does. Even figuring out evolutionary relationships.
How do you compare sequences? Is there a main tool for that?
Yeah, the workhorse is BLAST. Basic local alignment search tool. You feed it a sequence like one you just found, and BLAST scans these massive databases looking for similar sequences. It gives you an identity value, like how much percentage match there is, and an E-value.
E-value. What's that?
Expect value. It tells you the statistical significance. Like, how likely is it you'd find a match that good just by random chance? A low E-value means the match is probably real, evolutionarily meaningful.
Okay, so give me an example. How would that work in practice?
All right, say you sequence a chunk of DNA from a rat chromosome. You run it through BLAST against the mouse genome database. And bingo. You find a near-perfect match on mouse chromosome 8. It turns out to be the gene for the insulin receptor. It's 93% identical between rat and mouse.
So because you know what it does in the mouse.
You can be pretty confident it does the same thing in the rat. That's super powerful for figuring out gene function.
And this connects to homology, like related genes.
Exactly. Bioinformatics helps identify homologous genes. These are genes related by evolution. We talk about paralogs-related genes within the same species. Maybe they arose from a gene duplication event, like human alpha and beta globin genes.
Okay.
And these are homologous genes in different species. They usually have the same function, like the human LEP gene for leptin and the mouse LEP gene. They're over 85% identical. Finding the mouse ortholog really helped confirm the human gene's function in appetite control.
Got it. And all this labeling and identifying features on the genome that has a name, right?
Annotation. That's the whole process. You're essentially adding notes to the raw sequence, identifying gene regulatory sequences like promoter regions, maybe finding a TATA box, locating the open reading frames, the parts that actually code for protein with their start and stop signals, finding splice sites in eukaryotes, all the important landmarks.
So bioinformatics isn't just finding genes. It's understanding their context and potential function.
Precisely. It lets us infer function, understand evolutionary history, turn that mountain of raw data into actual biological insights. It's fundamental.
Which brings us back to the big one, the Human Genome Project, the HGP.
Yep. The culmination of a lot of these early developments.
Tell us about it. It was huge, right? International.
Absolutely. A coordinated international effort officially ran from 1990 to 2003. Big names involved, like James Watson initially, then Francis Collins leading it for much of the duration.
And the main goals, just sequence everything.
That was a huge part of it, yeah. Sequencing the 3 billion base pairs of the human genome, but also identifying all the human genes. The estimate back then was way off, actually.
How far off?
They were thinking maybe 80,000, even 100,000 protein coding genes. We'll get to what they actually found.
Okay, intrigue. What else were they aiming for?
Sequencing model organisms, too, like the mouse, fruit fly, yeast, developing new technologies for sequencing and analysis. And really importantly, setting up the ELSI program.
ELSI.
Ethical, legal, and social implications. They knew from the start this knowledge would raise huge societal questions. Privacy, discrimination, genetic determinism. They wanted to address those proactively.
That sounds forward-thinking. Now, wasn't there some kind of race? A private company involved?
Oh, yes. Genomics, led by Craig Ventergen. They jumped in using the whole genome shotgun sequencing approach, aiming to do it faster and, well, potentially profit from it.
Did that speed things up?
It definitely lit a fire under the public project. It pushed the HGP to adopt high-throughput WGS methods more aggressively. And ultimately, the public consortium actually finished slightly ahead of schedule, publishing a draft in 2001 and a more complete version in 2003.
So what were the big surprises? You mentioned the gene count estimate was wrong.
Hugely wrong. That was maybe the biggest shocker. Instead of 80,000 or 100,000 genes, the number turned out to be closer to 20,000 protein coding genes.
Only 20,000? That seems low. How is that possible given human complexity?
Great question. The answer lies largely in alternative splicing. Remember how eukaryotic genes have introns and exons?
Yeah, the non-coding bits get spliced out.
Right, but the splicing can happen in different ways. You can skip exons, use different splice sites. So a single gene, one stretch of DNA, can actually produce multiple different messenger RNAs and therefore multiple different proteins.
Ah, okay. So one gene doesn't just equal one protein.
Not even close for many genes. Turns out something like 94–95% of human genes can undergo alternative splicing. Plus, you have post-translational modifications. Proteins get chemically altered after they're made. So from those 20,000 genes, we probably make anywhere from 200,000 to maybe even 1 million different proteins. Complexity comes from versatility, not just numbers.
That makes more sense. What else did the HEP reveal?
Well, confirmation that only about 2% of our DNA actually codes for proteins. A huge amount is non-coding. Also, the incredible similarity between humans were 99.9% identical at the DNA level.
So the differences are tiny.
Relatively, yes. Most differences come down to single nucleotide polymorphisms, or SNPs, just single letter changes, and also copy number variations, CNVs, where larger chunks of DNA might be deleted or duplicated.
And the non-coding stuff, what's going on there?
A lot of it, over 50%, is repetitive DNA and transposable elements, bits of DNA that can kind of jump around the genome. And the sheer size of genes. The dystrophin gene, for example, is massive, 2.5 million base pairs long, but most of that is introns. Human genes are often much larger and more complex than, say, invertebrate genes.
So the HGP didn't just give us the sequence, it fundamentally changed our understanding of what a genome is and how it works.
Absolutely. It showed us how much we thought we knew versus what the data actually revealed. A real paradigm shift.
And it didn't stop there. The HGP really launched this whole omics revolution you mentioned earlier.
Exactly. It provided the foundation and the tools. Genomics led naturally to studying other large sets of biological molecules.
Like what? Give us some examples.
Well, proteomics, studying the entire set of proteins. Transcriptomics, looking at all the RNA molecules being expressed. Metabolomics, the set of metabolites. Glycomic sugars. Toxicogenomics, how toxins affect gene expression. Metagenomics, studying genomes from entire communities of microbes. Pharmacogenomics, how your genes affect drug response. The list goes on.
It's like looking at the cell or the organism from all these different molecular angles at once.
That's a great way to put it. And it also led to personal genome projects, or PGPs, sequencing individual people's complete diploid genomes.
Which leads to questions about clinical use, right? Like using WGS or WES for finding disease risks.
Yes, that's a big debate. Whole exome sequencing, WES, just sequences the protein coding bits, that 2%. It's cheaper, faster.
But you might miss things.
You definitely miss mutations in regulatory regions, promoters, enhancers, non-coding RNAs. Things that don't make protein but control how genes are used. WGS, whole genome, gets everything, but it's more expensive and generates way more data to analyze. There are pros and cons to both.
And these personal genomes are showing even more complexity.
They are. We're learning about things like somatic genome mosaicism, the idea that not all cells in your body are genetically identical.
Wait, really? I thought my DNA was the same everywhere.
Mostly, yes. But as your cells divide throughout life, tiny errors, mutations, creep in during DNA replication. So different tissues, even different cells within a tissue, can have slightly different genomes. It adds another layer to individuality.
Huh? Never thought about that.
And for things like bacteria, the whole idea of a single reference genome for a species is kind of dissolving. We now talk about the pangenome.
Pangenome.
It represents all the distinct genes and variations found across all strains of a species. Some genes are core, found in everyone, but many are variable, present only in some strains. It reflects the incredible diversity, especially in microbes.
And what about all that non-coding DNA, the 98%? Is it just junk after all?
Not so fast. That's where projects like ENCODE come in, Encyclopedia of DNA Elements.
What did ENCODE find?
It was a massive effort to figure out what all the non-coding parts of the human genome actually do. The big headline was, about 80% of the genome shows biochemical activity. It's involved in regulation. It gets transcribed into RNA. It's definitely not junk.
80%. So much for junk DNA.
Right. A lot of it gets transcribed into non-protein coding RNAs, like long non-coding RNAs or LNC RNAs.
Yeah.
There might even be more types of LNC RNAs than protein coding genes. They seem to play huge roles in gene regulation.
Wow. So the genome is way more active than we thought.
Absolutely. And we're still figuring out what it all does. There's nutrigenomics, how diet affects your genes, the Genome 10K project, aiming to sequence 10,000 vertebrate species.
And even looking back in time.
You bet. That's Stone Age genomics,
Huh?
analyzing ancient DNA. It's incredible what they can do now. They sequenced a 700,000-year-old horse genome from frozen bone.
700,000 years. DNA lasts that long?
Under the right conditions, yes. Frozen helps a lot. That horse DNA actually pushed back the evolutionary timeline for modern horses quite a bit. And they've sequenced woolly mammoths, found their, 98.5% identical to African elephants. It's just amazing.
That ancient DNA work is fascinating, and it ties perfectly into comparative genomics, right? Comparing genomes between species.
Exactly. Comparative genomics is all about looking at genomes side by side different species, sometimes even different individuals within a species, to understand genetics, evolution, and function.
What are the main uses?
Gene discovery is a big one. If you find a gene in humans whose function you don't know, but you find its ortholog in, say, yeast or flies where its function is known, that give you a huge clue. And developing model organisms for human diseases.
Because we share genes with them.
We share a surprising number of genes. By 2018, something like 23,000 whole genomes were sequenced. Humans share about 30% of our genes with yeast,
30% with yeast. Wow!
50% with fruit flies, 98% with chimpanzees, and maybe around 100 human genes even have orthologs in bacteria. It's powerful evidence for common ancestry and conserved core biological processes.
And the clinical relevance is there, too.
Absolutely. Roughly 60% of genes linked to about 300 human diseases have orthologs in Drosophila, the fruit fly. That's why flies are such valuable models for studying things like cancer, heart disease, neurological disorders.
It's not just the obvious models either, right? Didn't the sea urchin genome also show surprising connections?
It really did. Strongylus introitus preparatus. Its genome is about 814 million base pairs, 23,500 genes. And even though it's an invertebrate, way down on the evolutionary tree from us, it has some very vertebrate-like features. Nearly 1,000 genes for sensing light and odor, similar complexity to ours, and it shares orthologs with us for genes involved in human hearing, balance, and many disease pathways, like those involving protein kinases. We share about 7,000 orthologs overall with the sea urchin.
So these deep evolutionary connections are everywhere.
They really are.
Yeah.
It makes you think, if we're so similar genetically to so many other creatures, what really makes us uniquely human?
Which brings us to Neanderthals. That work must be shedding light on human uniqueness.
Definitely. The Neanderthal Genome Project, led by Svante Pääbo, who won a Nobel Prize for this work, by the way, has been revolutionary.
Sequencing DNA from ancient bones, how is that even possible?
Incredibly difficult. The DNA is degraded, fragmented, often contaminated. But they developed amazing techniques. They sequenced mitochondrial DNA first, then nuclear DNA. They even got DNA from a 400,000-year-old bone, the oldest Neanderthal DNA analyzed.
And what did they find? How similar are we?
About 99% identical at the DNA level. Very, very close. But comparative genomics let them pinpoint the differences. They found 78 protein coding sequences that changed in humans after we diverged from Neanderthals. Some are involved in cognitive development, skin physiology, sperm motility, hints at what might make us different.
What about language? There was that FOXP2 gene.
Right. FOXP2 is strongly linked to speech and language development in humans. Neanderthals had the same version of FOXP2 as we do. It doesn't prove they spoke like us, but it suggests they might have had complex vocal communication abilities.
But the biggest bombshell was interbreeding, wasn't it?
That was the shocker. The data showed clear evidence that non-African modern humans, Europeans, Asians, etc., carry about 1 to 4% Neanderthal DNA in their genomes.
So our ancestors met Neanderthals and had children.
It seems so. The thinking is these interactions happened somewhere in the Middle East, probably between 45,000 and 80,000 years ago, after Homo sapiens migrated out of Africa. It's rewriting the story of human evolution.
Incredible stuff. Okay, shifting gears a bit, from ancient humans to modern microbes, let's talk about metagenomics.
Right, metagenomics or environmental genomics. Instead of sequencing one organism, you sequence everything in an environmental sample.
Like what kind of sample?
Water, soil, air, sewage, the gut of an insect. You name it. You basically collect the sample, extract all the DNA from all the organisms present, mostly microbes, and sequence the whole mix using that shotgun approach.
And the big advantage is
you don't have to culture the organisms. Most microbes, maybe 99%, can't be grown in the lab using standard techniques. Metagenomics lets us see who's there and what genes they have, bypassing the need for culturing. It's revealed millions of previously unknown species and genes.
Any cool examples?
Craig Venter sailed his yacht around the world sampling ocean water. His expedition found over 1.2 million new DNA sequences, identified 148 unknown bacterial species, just from surface water.
Wow.
Or a study of the New York City subway system. They swapped surfaces everywhere, turnstiles, benches, poles.
What did they find? Anything scary?
Mostly common microbes found on human skin or in our gut, which makes sense. But here's the kicker. Nearly half of the DNA they sequenced didn't match anything in any known database.
Half. Just unknown life.
Unknown sequences, anyway. It highlights the vast amount of microbial diversity out there that we haven't characterized yet. It's often called microbial dark matter.
And this approach applies to us too, right? Our own microbes.
Absolutely. That's the Human Microbiome Project, or HMP, a huge effort to catalog all the microbes living on us and inside us.
What did the HMT find?
Well, they identified a huge percentage, 81 to 99% of the microbes and viruses at various body sites. Each of us carries up to 1,000 different bacterial strains. And the total number of microbial species across the human population is estimated at maybe 10,000.
Is there like a standard human microbiome?
That's a key finding.
Yeah.
There's no single reference human microbiome. It's highly personalized. It varies hugely between individuals, influenced by diet, genetics, environment, lifestyle, and it starts developing right at birth.
How is this useful clinically?
Hugely useful. Understanding the microbiome helps us figure out why antibiotics can sometimes cause problems by disrupting the normal gut flora, or why certain microbiome compositions might make people more susceptible to chronic diseases like inflammatory bowel disease, IBD, psoriasis, maybe even obesity or type 2 diabetes.
Can they link specific microbes to specific conditions?
They're starting to. For example, studies on acne found specific strains of the bacterium Propionibacterium acnes that seem to be strongly associated with having acne, while other strains might even be protective.
So it's not just which microbes, but which strains.
Exactly. It's getting much more detailed. They can even look at the combined microbial genes present in different diseases. Venn diagrams show unique gene sets associated with, say, liver cirrhosis versus type 2 diabetes versus IBS, but also some shared gene functions across them. It's complex, but incredibly informative.
Okay, so we have the genome blueprint, genomics, the environmental context, metagenomics. What about seeing the blueprint in action? Which genes are actually being used?
Now you're talking about transcriptomics. Studying the transcriptome, the complete set of RNA transcripts being produced by a cell or organism at a given moment.
Why is that important?
Because it tells you which genes are on or off and how active they are. This gives you massive insights into cell differentiation, why a liver cell is different from a neuron, how cells respond to signals, physiology, and disease mechanisms. Even though most cells have the same DNA, their RNA profiles, their transcriptomes, can be vastly different.
How do you measure the transcriptome?
For a long time, the main tool was DNA microarray analysis, or gene ships. These are little slides dotted with millions of tiny spots, each containing a known single-stranded DNA probe for a specific gene.
Okay.
You then isolate RNA from your sample, convert it into complementary DNA, cDNA, label it with a fluorescent dye, and wash it over the chip. The labeled cDNA will stick or hybridize to the spots corresponding to the genes that were active in your sample.
And the brightness tells you how active.
Exactly. The intensity of the fluorescence on each spot is proportional to how much RNA from that gene was present. It lets you measure the expression levels of thousands of genes all at once,
Right.
a snapshot of gene activity.
Is that still the main method?
It's still used, but increasingly RNA sequencing, or rna-seq, is taking over. It's a more modern approach using high-throughput sequencing.
How is RNA-seq different?
Instead of just measuring hybridization intensity on a chip, rna-seq directly sequences all the RNA molecules after converting them to cDNA. So you get the actual sequence data, which can reveal splice variants, novel transcripts, single nucleotide variations in the RNA itself. It's more comprehensive and quantitative.
And you can do it on smaller scales?
Yes. You can do RNA-seq in situ literally inside the cell to see where RNAs are located.
Right.
Or even on single cells. Single-cell RNA-seq is huge now, letting us see the variation in gene expression from cell to cell within a tissue, which you lose when you grind up the whole tissue. It lets you connect genetic variation and mRNA expression variation much more directly.
So transcriptomics shows the dynamic activity, not just the static code.
Perfectly put. It bridges the gap between the genome and what the cell is actually doing.
Okay, genome is the blueprint, transcriptome is the messages being sent.
Huh.
What about the actual workers, the proteins?
Right. That takes us to proteomics. The proteome is the entire set of proteins encoded by a genome or present in a cell or tissue at a specific time. Proteomics is the study of these proteins, identifying them, characterizing them, quantifying them.
What kind of information does proteomics give you?
All sorts of crucial stuff. Protein structure and function, of course,
Yeah.
but also post-translational modifications, those chemical changes made after translation. Protein interactions, who works with whom, where proteins are located in the cell, how stable they are, how long they last.
And this is where that gene count versus protein count discrepancy really hits home, right?
Exactly. Beadle and Tatum's old one gene, one enzyme idea. Genomics and proteomics blew that out of the water. Remember, 20,000 protein coding genes, but maybe 290,000 or more different proteins thanks to alternative splicing and post-translational modifications. The Human Proteome Map, HPM, is trying to catalog all of this.
How do you even begin to analyze that many proteins?
Well, a classic technique still used is two-dimensional gel electrophoresis, or 2DGE. It separates proteins in a complex mixture based on two different properties.
Which are?
First dimension separates them by their isoelectric point, basically, their overall electrical charge. Then the second dimension separates them by their molecular weight, their size. You end up with a gel showing potentially thousands of distinct spots, each spot ideally representing a different protein.
But how do you know what protein is in each spot?
That's the challenge. You have to cut the spot out of the gel and identify the protein. And the key technology for that nowadays is mass spectrometry, or MS.
How does mass spec work for proteins?
There are different ways, but a common one is MELD, matrix-assisted laser desorption ionization. You typically digest the protein from the gel spot into smaller peptide fragments. You mix these peptides with a special matrix chemical, put it on a target plate, and zap it with a laser.
Zap it.
Yeah. The laser vaporizes the matrix and ionizes the peptide fragments, gives them an electrical charge. These ions then fly through the mass spectrometer, and the instrument measures their mass-to-charge ratio very precisely.
And that tells you what the protein is.
It gives you a unique pattern of peptide masses, a peptide mass fingerprint. You can then compare this fingerprint against databases of known protein sequences, computationally digested into theoretical peptide fragments to identify the original protein in your spot. It's incredibly powerful for identifying the actual functional molecules of the cell.
This is all amazing, reading, analyzing, understanding existing life. But the final frontier seems to be designing it. Synthetic genomes.
That's definitely where things are heading.
Yeah.
It started with a really fundamental question. What's the minimum number of genes you actually need for a cell to live?
How do you figure that out?
Early work focused on bacteria with naturally small genomes, like Mycoplasma genitalium. It only has 525 genes to begin with. Researchers used techniques to systematically knock out genes one by one to see which ones were absolutely essential for survival under lab conditions.
And the answer was?
They estimated around 375 genes were essential for M. genitalium. Less than the full set, but still quite a few.
But then they went further. Actually built a genome.
Yes. That was the huge breakthrough from Craig Venter's Institute, JCVI, in 2010. They created the first functional synthetic genome.
How? Did they just type it out?
Almost. They chemically synthesized short pieces of DNA, cassettes, based on the known genome sequence of another bacterium, Mycoplasma mycoides. Then they painstakingly stitched these synthetic pieces together in yeast cells to assemble the entire 1.1 million base pair genome.
They built a whole bacterial chromosome from scratch.
Essentially, yes. And then came the really clever part. Genome transplantation. They took this synthetic M. mycoides genome and put it into a recipient cell of a different species, M. capricolum, whose own DNA had been removed or inactivated.
And what happened?
The synthetic genome took over. It rebooted the recipient cell. The cell started making M. mycoides proteins and took on its characteristics. Venter's analogy was like changing a Mac into a PC just by putting in new software. It showed that the DNA software really does define the cell.
That's incredible. Did they manage to make a minimal genome that way too?
They did in 2016. They designed and synthesized a stripped-down version called JCVI Syn 3.0. It has only 473 genes, the smallest genome of any self-replicating organism.
And do we know what all 473 genes do?
Here's the kicker. No. A surprising one-third of those absolutely essential genes have functions that are still unknown. We know the cell dies without them. We don't know why. It shows how much fundamental biology we still have to learn.
So the future is designing custom genomes using tools like CRISPR.
CRISPR-Cas definitely makes genome editing much easier and more precise than ever before, accelerating this whole field. And yes, people are thinking about custom design. The Human Genome Project, right, or HGP, right, was initially proposed to synthesize a whole human genome from scratch.
A synthetic human genome. That sounds ethically loaded.
Extremely. There was a lot of debate. The project has since scaled back its ambitions, focusing now on more specific goals, like engineering human cell lines to be completely resistant to all viruses, which would be huge for biomanufacturing. But the potential is there.
What are the potential applications of synthetic biology?
Oh, they're vast. engineering microbes to produce biofuels or pharmaceuticals more efficiently, designing bacteria that can clean up pollution, bioremediation, creating semi-synthetic crops engineered for drought resistance or enhanced nutrition. The possibilities are almost endless.
But it also raises those huge ethical questions again. Designing life. Where do we draw the line?
That is the question. As we gain this incredible power to read, write, and rewrite the code of life, we absolutely have to grapple with the ethical implications. Who gets to decide? What are the risks? How do we ensure equitable access?
And it's not just about future tech. Even the data we have now raises issues, right? Privacy.
Big time. That genetics, ethics, and society section in our source highlights this really well. The issue of genomic privacy is huge. Think about genetic testing companies, massive research databases.
Like that Ryan Kramer case, the boy who found his sperm donor father.
Exactly. He was 15. He used his own Y chromosome data from a genealogy company, combined it with some publicly available info, and managed to identify and contact his anonymous donor father back in 2005.
Wow. So re-identification is a real risk.
It absolutely is.
Yeah.
Your genome contains incredibly personal information, not just about disease risks, but potentially physical traits, ancestry. And once it's out there or linked to your name, you can't easily take it back. It raises concerns about genetic discrimination, unforeseen uses of the data.
So let me turn this to you, the listener, directly, as the source suggests. Would you send your DNA off to a private company? Would you get your whole genome sequenced? What kind of privacy guarantees would you need first?
It's something we all need to think about carefully as this technology becomes more common. What are we comfortable with?
Okay, we've covered a massive amount of ground here. So what does this all mean for you, the learner, the intensely curious mind trying to get a handle on this field?
Well, I hope you see the incredible journey we've been on. From Sanger sequencing that first tiny virus,
Through the massive Human Genome Project, the rise of bioinformatics making sense of the data deluge.
to the whole omics revolution, looking at RNA, proteins, microbes,
Yeah.
uncovering layers of complexity we never expected.
It really has transformed our understanding of life, hasn't it? From single genes right up to entire ecosystems and even our own evolutionary past with Neanderthals.
Absolutely. And think about the implications moving forward. We've seen how genetics informs our health-personalized microbiomes, affecting diseases like IBD, the potential for engineered cells, maybe even synthetic organisms doing useful tasks. How will these fields reshape medicine?
And maybe even reshape our definition of what it means to be alive, how we understand ourselves.
That's the really deep question, isn't it? The journey of discovery here is definitely far from over. It's an incredibly exciting time to be learning about this stuff.
Couldn't agree more. Well, thank you for joining us on this deep dive into the truly fascinating world of genomics, bioinformatics, and proteomics. We really hope this gave you a powerful shortcut to being well-informed.
We hope it sparked your curiosity to keep exploring.
And thank you, as always, for being part of our last-minute lecture family. Until next time, keep exploring.